Hello everyone, this is my presentation on the antibody prototype, and this is my antibody. Right here on this first slide I just have a picture of what a typical antibody looks like. Make it a little bit bigger so you can see. So the heavy chain, and then the light chain, then we have the hinge region here in the middle, the antigen binding site, and then we have the disulfide bonds here in the middle of the hinge region, and then I'll go through and talk about each one separately. So starting with the heavy chain, this is the largest of the two chains, signified by the blue clay right here. There's two on the antibody molecule. There are 5 different types of heavy chains. It's classified as the IgH gene and is located on chromosome 14. It has 1 variable domain and 1 constant domain. Total amino acids it's made up of is about 450 to 550, but each variable chain is made up of about 110 amino acids each. The light chain is the smallest of the two chains, signified by the red clay right here. It has 2 types. Kappa and lambda is classified as the IgK and IgL genes. It is located on chromosomes 2 and 22. It also has 1 variable domain and 1 constant domain, and it is made up of about 211 to 217 amino acids. The peritope is the antigen binding site of the antibody, and it is signified by the green clay up here at the top. So it comprises both parts of the heavy and the light chains, so it connects to both of them, and it is located right at the tip of the antibody on the antigen binding fragment, which is right below this. The hinge region is going to be this little yellow area in the middle, and this is located between the FAB and FC portions, and it is a stretch of the heavy chains, so this is how they are connected, is by the hinge region. This unique structure allows its position to be flexible and have the segments move in different ways, so that way it can bind to different antigens better, because some might be wider or smaller, so this way it allows it to kind of move so that it can better bind to specific antigens. The disulfide bonds are going to be signified by this little orange strip right in the middle of the hinge region, and since antibodies are able to function in oxidative extracellular environments, a disulfide bond is formed when a pair of cysteines oxidatively reacts in close proximity with one another. So I should give you a little close-up of my antibody. So again, we have our heavy chain, we have our light chain, we have our hinge region, we have our disulfide bonds, and then we have the peritope here at the top. So I hope you like my antibody. Thank you.